Hello, everyone, and welcome back to Psychedelic Vantage. Today, I have the pleasure of interviewing Dr. Chris Witowski, co-founder and CEO of Psilera. Salera is an innovative biotechnology company designing first and best-in-class therapeutics for the treatment of neurological and neurodegenerative conditions. The company's lead compound, SIL-006, is a next-generation psilocybin derivative with a broad applicability and a patient-friendly profile. Because of its non-hallucinogenic profile, SIL-006 is designed to be a good candidate for take-home use and insurability. The reason I'm very excited to have Chris on the channel today is because of Psilera's focus on treating neurodegenerative conditions. I see it as something that really maybe puts them in their own league when it comes to comparing them to the many other companies in neuropsychiatry who are mostly focused on looking at non-hallucinogenic neuroplastogens for depression and anxiety. With that said, Psilera is also looking to research their compound SIL-025 for anxiety disorders. Chris, thank you so much for coming on to the channel today. How are you doing?
Doing well. Thanks for the intro. Glad to be here.
Absolutely. So before we get started, can you speak a bit about your background and how Psilera got started?
Sure. So, my background is in natural products chemistry. I got my PhD at the University of South Florida under Bill Baker, really a world-renowned natural products chemist. And what you do in natural products is you find new starting points for drug development by discovering new drugs from nature. About 70% of all drugs, FDA-approved drugs, start out as a natural product. Then it's companies like Salera that optimize them into better drugs, and ultimately, it's a very rich starting point. And as we're speaking now about psychedelics, I mean, many of these molecules are natural products.
Yeah.
And actually, that's where I met the other co-founder of Psilera, Dr. Jackie Von Salm. Actually did our PhD studies together. And, it was about 2019, not long after Spravato was approved, that we really, understood this was the right time to get into psychedelic research, neuroplastogen research. And together we had been doing drug discovery, drug development, formulation development, and held leadership roles prior at CNS companies for close to 30 years combined.
Oh, wow.
So it was really just kind of being at the right place at the right time, and using our backgrounds, which are really uniquely suited for the research that we're doing today.
That's awesome. Thank you so much for the background there. So earlier this year, Psilera announced the selection of behavioral variant frontotemporal dementia as the lead indication for its drug candidate SIL-006. So what is behavioral variant frontotemporal dementia? How prevalent is this condition, and how does someone get this condition?
Well, let's start first with frontotemporal dementia. First
Okay.
of all, it is a rare disease. And in these, you don't always have all the answers. I mean, what we do know is there is a genetic link to it, but like most dementias, that doesn't account for all patients. Only about 50% have a known genetic mutation that leads to FTD. With FTD, there are some really high-profile cases out there, such as Bruce Willis, who was diagnosed in the last few years, Wendy Williams, who really is showing some of these severe psychiatric side, which are common with BVFTD. And I would think, one of the most, use cases is actually Robin Williams. So he actually had a Lewy body dementia. He took his own life. And I mean, his late wife really summarized it well in an article she penned in the Journal of Neurology titled "The Terrorist Inside My Husband's Brain." And with FTD, it affects about 60,000 patients within the United States, roughly about 1 million worldwide. So for an orphan or rare disease, it is fairly common.
Okay.
It affects people generally younger than most dementias. So typically under the age of 65, it's the most prevalent or second most prevalent form of dementia. And patients can be diagnosed as young as 45. And typically what you see is really a psychiatric onset. So these patients start to have personality changes, mood swings, really violent outbursts, even changes in eating patterns or drinking patterns can be really disrupted. And about 50% of all FTD patients have this behavioral variant. And really, that's why we think 006 being this sort of next generation neuroplastogen is really uniquely suited for these patients with these severe psychiatric issues.
That was a great overview. Thank you so much. And before we were doing this interview, I was trying to learn a little bit more about the condition, and just there was a story of just some a family member of someone who has it, just explaining just how devastating it was, not only for the person suffering with the condition, but also the family. Like, for example, in this case, this person, I think, was actually 45, in the prime of their life, amazing job. And then all of a sudden he started to, show a loss of interest for the family, and it'll maybe the lack, the not being able to empathize properly and acting out. So it seems like a very devastating condition, and I'm so happy to hear that Psilera is trying to tackle this condition. So, with all this said, what treatment options are there now for FTD or behavioral variant frontotemporal dementia?
Currently, there are no approved treatments for FTD, for BVFTD. So these patients really have no options. Some physicians do prescribe off-label SSRIs or antidepressants to kind of cope with the symptoms. What I can say, there have been multiple clinical trials with these SSRIs, and they provide no clinical benefit to patient. And something you said is so true, that it just doesn't affect the patient. I mean, now these caregivers are having to really, change their lives, spend a lot more time with them. It is really devastating for the families, for the caregivers of the people with FTD. And again, I mean, it affects people younger in their life. Like you said, 45. I mean, these are people in the prime of their careers, and to have it all ripped away and without any available treatments with any benefit is one of the reasons we really looked at FTD, not least because of the science that we're developing and why we think it's so uniquely positioned there.
So, before we get into maybe how SIL-006 was discovered, I guess the goal here with SIL-006, is it to slow down the progression of FTD? Can it potentially reverse it? Or also, can it help manage symptoms?
Our goal is really to show all of the above.
Oh, wow.
Where we're starting right now is looking at the symptoms, the psychiatric issues. Apathy is a really prominent feature in these patients, and
Yeah.
we're working with world-renowned researchers now in phase 2 trials, their own trials to validate some of these endpoints. And what we've shown with our drug, at least in preclinical models, is it works very rapidly. Within 24 hours, we're showing a noted antidepressant effect. We're also showing better sleep patterns, better procognitive ability, and some of the compulsions that are also associated with FTD as well. So we're not just covering the symptoms. We also think the way the drug works as a neuroplastogen is really unique. Of course, with any dementia, including FTD, you have atrophied neurons that are dying. Your brain is shrinking, and specifically in FTD, as the name suggests, it's in the fronto part of your prefrontal cortex. And that's exactly the emotion center of the brain. And that's why things start to go haywire for these patients. But also, when you look at the main drug target, or one of the targets for neuroplastogens, including 006, is 5-HT2A. It's a serotonin receptor. It's responsible for neuroplasticity, number one, but also, depending on your drug, can also tip it into hallucinogenic effects, which is where, psychedelics are active. The interesting thing about 006 is it targets 2A in the prefrontal cortex. We've demonstrated this in animals, and 5-HT2A is dysregulated in patients. So they have a lower density of these receptors. And as well, we've shown inflammation markers in the brain too, which are also quite prevalent in FTD along with other dementias. So the way the drug works, we think, can actually slow or reverse the disease. But
Wow.
initially, we're starting at a symptomatic treatment because we can demonstrate this benefit very rapidly given how the drug works. And then once we prove this concept in clinical trials, we will extend it out further to see if there's any benefit to the underlying disease, to cognition, through other endpoints.
Wow, that's absolutely incredible to hear and really super fascinating to listen to you explain this. So I guess can you maybe tell us a bit about how SIL 006 specifically was discovered?
Yeah, I would love to. So as I mentioned, myself and the co-founder, Jackie, both are natural products chemists. So we've been looking at this space for a long time. And, we really had some unique insights on how to make small modifications to serotonin-like drugs, including psilocybin and DMT, both serotonergic compounds. So we started there as sort of an initial thought pattern. And from there, we've developed a platform, which we call the Salera Third Eye.
Mm,
And what this does is combine computational chemistry. So I'll use some hand models here in a minute just to
beautiful.
better explain it. But really, in a sense, what it is a drug binds to a receptor or a protein. So I mentioned 5-HT2A. It's a very well-known receptor. There's about 20 or so drugs that have been approved as 5-HT2A modulators, whether they
Okay.
be agonist, antagonist, some effect on 2A. And they've also been crystallized with the drug intact. So when you look at a drug, you've got your active site. I'm raising my hand, and you have your drug that fits within there, and it has a number of interactions that allow the drug to bind. Well, what we can do using computers is now take out the drug from the known receptors and now create our own drugs and fit them in there. So we had a really rich library of known non-hallucinogenic 2A drugs, as well as known psychedelics, 2A hallucinogens. So from there, we were able to create a really rapid model and develop. I mean, we screened hundreds of thousands of compounds in a few months. Really, what that allowed us to do is develop a patent portfolio, which we now have issued on millions of new neuroplastogens. And we can build these drugs atom by atom to make them purpose fit for the molecule or for the receptor of interest, reducing hallucinogenic effects, reducing some of the other side effects, cardiotoxicities associated with other serotonergic receptors. And of course, we developed this in-house with a great team of computational chemists. We now have medicinal chemists who've made hundreds of these drugs, tested them in assays with receptors and animals to validate what we're seeing through our platform. So really what I can share is our predictability from making these drugs in computers to what we're seeing in the real world is, I mean, we haven't made a drug yet that was not as we predicted via our platform. So it's really allowed us to optimize
Wow.
these very quickly and develop a really, 6 new classes of neuroplastogens. So yeah, testament to the team for sure in some of this work. But through that discovery program was how we found SIL 006.
That's incredible. And thank you for explaining and congratulations on the progress. I guess maybe a little bit of a side question here, but I would be curious. So 006 is a psilocybin derivative. Are you looking at other psychedelics to inspire, I guess, these non-hallucinogenic versions, maybe 5-MeO-DMT, NN-DMT, maybe ibogaine?
Yeah, I mean, great question. So, I mean, let's take 5-methoxy-DMT. I mean, really, that's 2 methyl groups away from serotonin.
Hmm.
And of course, all these molecules work on the serotonin system. So, really for us, it's a serotonergic type application. So, we have derivatives of 5-methoxy-DMT. The triptan drug class is well known for headaches. There's been dozens of approvals there, making new derivatives of that, psilocybin, DMT. I mean, there's really a few points on the molecule that you can add even just a single atom and completely change its profile to reduce hallucinations. So, we're not going as complicated yet as ibogaine, just because,
Yeah.
there are some underlying issues with that molecule,
Yep.
cardiovascular issues that, we're really sticking with drugs like psilocybin, which are very well known. They've been dosed in hundreds, probably at this point, thousands of patients in clinical trials, very few side effects other than hallucinations, which we found are pretty easy to reduce just with a single atom.
I see. And is 006 eligible for orphan drug designation for BVFTD? And if so, what are the implications of this for Salera and potential future patients?
Yeah, great question. So orphan drug designation, you can do this for a treatment which affects less than 200,000 patients within the United States, and there are no available treatment for those patients. So FTD does fit this criteria. We are actively discussing with the FDA now an orphan drug designation.
Hmm.
What I can tell you is some of the interactions is BVFTD is a sub indication of FTD. So we are going to target the main FTD population, a larger population of about 60,000 in the US, just because any sub indication under that could potentially benefit from our drug. So we're certainly working towards that. Hopefully, we'll have some news to share soon. But certainly, that is a path that we're investigating because there are significant benefits to a drug developer like us. Um, First of all, you get more interactions with the FDA, which is, of course, important when you're developing a new drug in general. You also can get things such as breakthrough designation once you're in clinical trials. So that allows you to accelerate the development of your program. And for orphan diseases, generally, since there are no other drugs approved, I would say the median price for an orphan drug is about 200,000 per year per patient. So
Hmm.
there are benefits because you're going into a model where there's a limited patient population, there are no approved drugs, so there are benefits to go commercially by this pathway.
Okay, I see. Again, thank you for the overview there and explaining that really well to myself and the viewers. Now I'd like to move on, I guess, to maybe about neuroplastogens in general. And I'm sure you can touch on maybe what separates yours from other companies. But I guess overall, can you explain to myself and the viewers maybe the difference between how microdosing may work and how non-hallucinogenic neuroplastogens might work?
It's a great question for sure. So with neuroplastogens, I'll start there. So these are drugs which at no dose would be hallucinogenic. I mean, we've trialed
Okay.
lots of doses, very high doses in animals, and haven't seen anything that resembles a hallucination or psychedelic like effect in these animals. So with microdosing, yeah, you're just dosing a low dose of a psychedelic, so you're not tipping the scales into hallucinogenic effects. So what we've shown through some of our research and with collaborators is there's definitely think of it as a gauge, right? I'll use some more hand gestures. So, um, with still 006, we're kind of right in the middle where if you go too far here, you're going to be hallucinogenic. If you go too far here, you're basically an antipsychotic, which, there are lots of those on the market. So with SIL-006, we are benefiting from neuroplasticity from the 5-HT2A effects, but we're not doing that via hallucinogenic pathway.
Okay,
And with known, we'll say psilocybin and microdosing, you're already tipping the scales. You're just at a lower dose.
I
And
see.
I would say for microdosing, of course, the clinical or blinded evidence for efficacy of microdosing is still to be determined.
Mhm.
So our belief is that if you can develop new drugs, number one, that's helpful for developing an IP portfolio, but as well, you don't have to worry about potentially using too much of a drug and then tipping into the side effects that you don't want in the first place. And from the research that's out there on microdosing, you are seeing some benefits to creativity, but you're not really seeing benefits of neuroplasticity. I believe the only preclinical study that's been published so far is showing benefit of microdosing DMT that actually
Hmm.
promotes plasticity. Actually, most of the other psychedelics at low doses, I don't believe have any neuroplastic effects.
Was that study from David Olson, if I'm not mistaken?
I believe so.
Very interesting, and I'm so happy you were able to explain this because we did have another company on the channel. I can't think of the name right now, but they're working on non-hallucinogenic neuroplastogens. When I asked him that question, I had no, I didn't understand his answer, so I really appreciate. I think I have a much better understanding of maybe the differences here. So, um, I guess this is an interesting question. I think. Like what type of dosing regimen is Psilera looking to test out? And the reason I ask is because I know some companies in this area of drug development are exploring or at least talking about maybe the pill being once monthly, and others are looking at more chronic everyday dosing. What is Psilera looking to do here?
With SIL-006, the first thing I'll point out is it's an orally available drug, about 60% oral bioavailability, which is great. As we're targeting an at-home patient use, we think an oral administration, it's just like any other drug that's out there. What I can say through some of our preclinical models is it's indication dependent, at least for SIL-006. So take psychiatry, for instance, and some of the anxiolytic or antidepressant effects. So we show a single dose is effective and it works very rapidly. So, potentially you're looking at more of a maybe an intermittent dosing regimen rather than a chronic dosing regimen.
Hmm.
Um, in other trials, we have done alcohol use disorder, for instance. So we paired SIL-006 alongside psilocybin and got animals addicted to alcohol. And over 60 days, we dosed these drugs every 3 days. So essentially similar to a maybe microdose regimen, but it was a full hallucinogenic dose of psilocybin, which in phase 2 trials of 100 patients, I think there was a 70% remission rate for patients with alcoholism. So
Hmm.
over that 60-day period, every third day, SIL-006 actually outperformed psilocybin, again, an intermittent dosing
Yeah.
regimen. Um, from what we've seen and other benefits, we think certainly what we're targeting is at least a once daily, or sorry, at most a once daily dose. We think ultimately we can formulate a drug that's in a once weekly format. There are many other drugs, maybe in a longer lasting injectable format that you can do in a once monthly. Once you have an effective and safe drug, there's a lot of things you can do to formulate a drug to make patient compliance and dosing easy. But all the underlying properties that we were looking for SIL-006, they're there. So that's a great starting point for us to now really validate what we're seeing in the clinic.
And for the viewers to know, what stage of development is 006 in right now? Perhaps when can we maybe see it enter a phase 1 trial?
Yeah, so right now we are in IND enabling safety and toxicology studies. So this is preclinical, but these are the last steps to prove that your
Hmm.
drug is safe enough to begin dosing in humans. What we expect is to submit our IND investigational new drug filing beginning of next year and begin our phase 1 study in healthy volunteers next year.
Wow.
And because we are going after FTD, this rare disease, we do anticipate to do a double arm, maybe 1B trial, where we're incorporating FTD patients into our phase 1 design so that we can begin to show the benefits of the drug in this patient population earlier than maybe most would be able to.
Very exciting. So as far as I'm aware, you are one of the only companies in the psychedelic slash neuroplastogen space whose lead indication that they are trying to tackle is a neurodegenerative disease. So why did Salera choose to go down this route?
It's a great question. And, from day one, when we created the company, we were always going to look in areas where there are huge unmet needs. And I think neurodegeneration, dementia, frontotemporal dementia, all of the above really, there's 6 million people in the US with dementia. And frankly, the approved drugs that are out there now, they, some would argue they cause more harm than good.
Wow.
Um, and number one, there's a huge unmet need, and especially for FTD, there's nothing for these patients.
Yeah.
And one of the reasons we really focused in this category is we're developing these neuroplastogens, so drugs without hallucinations. And we really think it opens up this patient population that otherwise couldn't benefit from a traditional psychedelic.
Hmm.
So these patients say with FTD, they have cognitive issues, they have issues with memory, some even have aphasia or communication problems. So how you can, let's say you dose a patient with a psychedelic and they're now integration process, they don't know where they are, they're violent. There's a lot of issues with dosing patients with neurodegeneration with the psychedelic. And as well, about 50% of all dementia patients have a high propensity for psychosis. So you're now giving them a psychedelic derived from psychosis. So, we really think these two things are really limiting for the current standards of care, the current drugs that are out there for these patients. So we think by removing these side effects, we really now can find new effective treatments in neurodegeneration. And, for the reasons that I mentioned, neuroplasticity is a really interesting mechanism that has yet to be trialed in patients with dementia, just because their brains are atrophying, their neurons are dying. And one thing we do know about neuroplastogens, they stimulate these neurons. They can aid damaged neurons. They can even create neurogenesis, create new connections. And as just a hypothesis, that's really something that needs to be trialed.
Yeah.
And of course, the way our drug works very quickly with psychiatric issues, we think this can provide a symptomatic benefit to patients. But again, longer term, we really think it can be disease modifying for these patients. And the last thing I will point out about SIL006 is its mechanism is a dual agonist of 5-HT2A, the serotonin 2A, as well as 5-HT6. And 5-HT6 is known in terms of literature for about 20 years to being strongly linked to cognition and memory. And it has been trialed all the way to phase 3 in Alzheimer's disease. Like many treatments, those other ones didn't work out. We found a new way of stimulating SIL006 through agonism. So really, SIL006 will be the first 5-HT6 agonist in clinical development. And, we really think this is going to provide a new option for patients all the way from symptoms to potentially better quality of life for themselves, for their caregivers. And we really look at this as kind of the tip of the spear. We really think there is a lot of potential for neuroplastogens in dementias and other neurodegenerative diseases.
I can't wait to follow the progress in the coming months and years. And I actually wasn't aware how close you guys were to getting into a phase 1. That's really exciting. So from looking at Psilera's website, I noticed the company is looking at, and we did touch on it a little bit, is looking at other next generation psychedelic treatments. At least I think they're based on psychedelics. Can you speak on these other compounds Psilera is working with?
Yeah, so as I mentioned in some of our platform work early on in drug discovery and some of the work we've been doing internally, we've developed 6 new classes of neuroplastogens. So SIL006 is in its own class, but we also have 2 others that we're actively developing in. One of these categories, as you mentioned, is called SIL025. In this class, I would say from a structural standpoint, maybe more similarly related to 5-methoxy DMT. And what we are seeing, at least in preclinical models, is really rapid anxiolytic or reductions of anxiety.
Yeah.
The drugs that we've made so far look really promising, really stable, great safety profile. Of course, non-hallucinogenic. Everything that we're developing is a neuroplastogen without psychedelic or hallucinogenic effects. So, these are a little bit earlier stage than 006.
Hmm.
And we also have SIL020, again, in its own class of serotonin-like molecules. And what we see here, again, is strong pro-cognitive effects. So improvements in learning and memory. We also see, particularly with 020, actually good fear responses. So similar to something like PTSD.
Mm.
These are a little bit earlier stage, so we're not going to comment exactly on where we're going to position them from a therapeutic profile. But as we see with 006, I mean, there really is a lot of promise across many CNS disorders with these types of molecules.
And and again, you did touch on it about 006, but I guess maybe overall as a company, what are the next steps for Psilera moving forward? What are you looking forward to?
So as I mentioned, we're in IND enabling studies showing our SIL006 is safe in multiple animal species with multiple doses. What I can share so far is it looks very safe. We have a great therapeutic profile. So we fully expect we'll be submitting an IND in the next few months ahead. And this is a huge inflection point for a company like Psilera
Mm.
in going from a preclinical discovery company into a clinical development company. And, really excited for this transition. So far we've filed, I believe, 17 or so patents to date covering new formulations of known psychedelics, a lot of our neuroplastogen development and discoveries in there. We have 3 issued patents covering all of our work. We expect several more of these in the next few months. And it's with patents, it's an ongoing strategy. But we started the company in 2019 before really, I would say, the industry really took off.
I
So
see.
that put us in a position to develop a very strong patent portfolio. The drugs we're developing are differentiated from other programs. And as you mentioned, with the orphan drug status, that is something that we're actively pursuing. And you don't need clinical evidence in order to file or get acceptance for an orphan drug designation. So, I mean, really, in the next 12 months, it's going to be a very exciting time for Psilera, and I'm really excited.
I'll be looking for those press releases. I know for sure I'll be looking out for those. Very exciting. Now to wrap up here, what excites you the most about the future of psychedelic medicine? I know you follow this area as well outside of the non-hallucinogenic neuroplastogens. So what excites you the most about that? Again, non-hallucinogenic neuroplastogens. And is there anything that worries you or maybe keeps you up at night about the space?
I mean, I'll start with why I'm excited. I mean, number one, in the 1970s, if we didn't ban these molecules, who knows where we'd be today? Because, I mean, you look at the timeline, 1970s, they banned psychedelics, 1980s, SSRIs are approved. And most pharmaceutical companies just kind of said, all right, we cured depression. Clearly, we know that's not the case. You talk to many of these major pharmaceutical companies, as we have now, and they say this era of neuroplastogens, specifically those without hallucinations, those are really the only area they're interested in
Mm.
terms of partnering, developing. Many of these major companies have said, I mean, this is going to be a new era of we call it mindful medicine, but of new CNS drugs, neuroscience practices. I mean, we just have much better tools now to understand how these drugs work that we didn't have 50, 60 years ago. So that gives us better ways to develop these, maybe with fewer side effects, such as neuroplastogens. And I think what we're seeing today is really just the tip of the spear for what's possible. I mean, as I mentioned, a lot of the development now is in psychiatry, which is great because there is still an unmet need there. But I think the potential in neurodegenerative diseases like FTD, like dementia, it's really exciting. Even 50% of all dementia patients have severe psychiatric issues, psychosis, depression, anxiety. So, I mean, there's still a lot that needs to be developed in order to provide new standards of care for patients and to develop things that work very rapidly. You don't have to wait 4 to 6 weeks in order for
Mm,
them to show any benefit. And even then, 30 to 40% of patients don't respond.
yeah.
I mean, to me, that is the most exciting. And to be able to be on the forefront of this development, um, sometimes I kind of have to pinch myself because, I mean, this is, I'm going to spend the rest of my career here for sure. And I
Wow.
mean, in the next 5, 10, 20 years, I think you're going to see an entirely different CNS treatment landscape, 100%. Psychedelics will be approved. I mean, of course, there's been some setbacks recently, but the amount of evidence that's out there, um, it's not a matter of if, it's a matter of when. And of course, these neuroplastogens, building off the existing knowledge that we have and
Mm,
just reducing some side effects. I mean, this isn't a new concept. This is what happens in every drug development target. You find one target that works, and now you figure out, okay, how do you make the drug better? How do you reduce
mm,
side effects? How do you make it orally available so you don't have to have an injectable? So you're definitely going to see a stepwise process, over the next, even few years for sure.
mm.
To your question on what keeps me up at night, hmm. Well, I mean, it's obviously been a challenging time for the industry. Of course, there's been ups and downs, which it's common with any industry emerging with the stigma. I think that adds a little bit of extra um, friction to the industry, I would say, especially from uh pharma companies for sure. I mean, you're now starting to see some of them work in the space, whether it be directly with licensing agreements, through
Mm,
investments even. So you're starting to see the scales tip. And frankly, CNS is in since the 1980s, CNS has not been the area you want to be in drug development. But now we are seeing the tide shift. Oncology had its heyday for the last few decades, but you're really starting to see some new things happen in CNS. I mean, for instance, I'll name drop Bristol Myers Squibb. They haven't been in CNS for decades, and now they acquired a company for 14 billion. And you're now starting to see some older, the legacy CNS operators really be interested in the space. You're seeing people who had no interest at all now
mm.
obviously very interested because of the unmet need, the emerging science. So yeah, I mean, it's, of course, with any industry you're going to have ups and downs, but we're taking a long-term view because we know the science is there. It's just a matter again of when, not if.
Mm. Really, really well said. Listen, Chris, thank you so much for coming on the channel. It was great having you on. Um, and everyone else watching this, thank you so much. If you enjoy this, please like, share, and I hope you have a great day. Thank you, Chris.
Thank you.